I wanted to say, so,
yeah.
so I overheard that you were like, oh, this is like really fast paced. So you don't expect to understand each piece of code that we present. We just like put all information that we have on you, and then once you have time and you need different pieces of analysis, you sit down and go through them, but you have like in the back of your head, you have all the possibilities that there are, and then to actually find out how to do it, you go back to the code, go back to the presentations and figure it out for yourself. Don't expect to understand and to keep everything that we're presenting. It's way too much information, but the aim is just that you have all the information kind of in the back of your head, that you know it exists, and that you know something like, there was something that I should have to take into account, and then you can go back to the scripts and the presentations, just so you don't to lower your expectations of yourself.
Yeah, I think this is very important, and also it's important that you ask questions, especially about the concepts, because then those are those that we really would like you to bring home, and then when it's about the code, then of course other people might help you, or you will find information online, or there is a lot of resources. But I think the concepts is what you really should focus on, and then also here we will present in this all week, and also all the other tutors, so many methods. I personally didn't use all of them in like 8 years. So you won't need all of these probably. You will just see all that is available and then try to pick what is interesting for you and for your analysis, and then you will become very good at that thing. But like all of this is just for you to know what exists, what is possible, and of course there is way more possible that we are not even covering, but just to get you familiar with what you should have in the back of your head when you analyze data, what to be aware of, what you should be careful about, and so on. But then, yeah, you'll be fine. Okay, so today we continue talking about how to analyze movement without context. So we're still, again, not talking about environmental data yet. We will do like just a brief example at the end of the morning. But in general, today we're going to focus on what we call trajectory-centered analysis. And I will present a few methods that are very useful to understand or to quantify what is the movement process that the animal is going through. And to do this, there are a couple of things to keep in mind. Half, I think, of the methods that I will present are really scale sensitive and sensitive to the sampling schedule because they are based on distance. So this is all that we talked about yesterday, so this is something you have to be aware of. And also some of these methods need an equidistant projection. So again, repeating what we talked about yesterday and the different type of projection. So for some of the method, you are required to reproject your data first. Autocorrelation. This also, we mentioned it yesterday, and it's kind of intrinsic to movement. So whenever an animal is moving, there is some degree of persistence in the type of movement the animal is doing. So if I'm going very fast, most likely I'm going to go very fast for the next certain amount of time. If I'm going very straight, the same. There is also some intrinsic correlation between how fast I go and how straight I go. And so all these things are in the data. And so we see them in the data, and they're just part of how the movement is working. Autocorrelation is basically the correlation, the degree of correlation of a variable with itself, with a metric with itself, so speed with the speed, azimuth with the azimuth, and so on, at a certain lag of time. So if I'm going very straight now, how likely it is that I'm going to go straight the next step, and then in 2 steps, in 3 steps, in 4 steps, and so on. And so this is very useful to understand what is the movement process that is going on. And here there is a simple function that we'll see later in R that does this for you, autocorrelation function, and this is just an example of how the autocorrelation is in the speed of Fisher. And then you see that for the first 7, 8 observation, there is some degree of autocorrelation in the speed. Of course, how the interpretation of this depends on the sampling frequency. In this case, I don't remember how often were the data collected, 10 minutes maybe? So I was saying the correlation structure informs us about the movement process. So if we look at the correlation in azimuth, for instance, azimuth was the direction of movement. So if I'm going in that direction and the next step I keep being that direction, then you see in the plot on the left that I go far away from my point of start very quickly, because I just keep straight and I go away very fast. If my correlation in azimuth is very low, then this means that my likelihood to turn around and change my direction is very high at every step. And so it means that I'm not going to go away very fast, but I'm more likely to meander around and forage around and stay longer in a certain space. So how do we quantify that? So because now we went the other way around, we are looking at the correlation process and how this affects the movement. But once we just see our trajectory, how do we quantify it so that we know what the underlying movement process was? And there are different methods that I will show you today. We have the net square displacement that basically quantifies what is the distance. I will show in a bit with more detail, but it shows across the trajectory how far away I'm going to go each step of my trajectory from a point of reference. That could be my nest or the first point of the track and so on. And the first passage time does something similar, but when I don't have a point of reference, then I will look at this throughout my trajectory. So for each point, each location in my trajectory, how long does it take to cross a certain radius? And there will be parts in the trajectory in which I'm going very directed, and so the time that it takes to cover that part is very short, but then other parts in which I'm slow, and then the time that will take to pass it, it's longer. And then we will look at recursions that use very similar tools, but then they will look at how often a certain location is revisited. So this is very useful very often when we are interested in revisitation of a resource or a nest and so on. And then finally, we will look at the variance components of movement. And this is something that also came up yesterday with one of the questions. And it's basically how, if I don't want to interpolate, what is a better way to have an estimation of the probability of encountering an animal where I did not observe it. So I know I have my steps, my observations, and then I can have shorter or longer gaps in between observation. So how can I have some sort of estimation of where the animal could have been in those gaps, given the animal is moving in a certain way? So we'll go step by step and show some code on the way. We will start with the net square displacement. So the net square displacement calculates the distance from each location in our trajectory from a reference point. Here, the reference point is showed as the first point of the trajectory, but it could be anything else that we decide. And the way to calculate it is very, very easy. Any package that calculates distances, basically, you can do it, for instance, with SF, because then you also can leave it in spherical coordinates. So in this case, you don't need to reproject. You can calculate the distance of each point in the trajectory from a reference point. And this is very interesting if the reference point has a meaning for you, like the nest or something else. So here we already talked about Leo the vulture yesterday. We saw that Leo was migrating for several years, and Leo was tagged in the nest, so the first point of the trajectory, in this case, it's the nest. And then we can see how over time, the net square distance first is very low when the animal still is in the nest, and then it goes very far away. Then it jitters around. This is probably the wintering ground. Then the animal migrates back. And this might not look like this, but for this animal, we are lucky because Leo every year sits basically in the same nest or very close to its nest. So this is the distance from the first point of the trajectory is exactly the same every single year. This might look a bit different for different species, but still you would see that there is some going away from if they maintain the same breeding ground, there will be some going away from the breeding ground and some coming back every year. And so it's very handy if we want to classify what's happening in the trajectory, and we want to focus, for instance, only on the wintering, only on the migration time, and so on. And you will see more about this also later with segmentation. First passage time. Most often, unfortunately, we don't know what is an interesting reference point for our trajectory. First time we're looking at this data. So one option we have is that we're going to use each location in the trajectory as a reference point. And then we decide on a radius, or usually on many radii, because we are interested in looking the difference of using different radii. And we're going to draw a radius around each of the location. Sorry, I go back to this because I wanted to say, yes, there is this package that helps us calculating it. And in this case, I will tell you why regular sampling it's important, and also equidistance projection. But so what we do is that we draw a circle of a certain radius around each location of our trajectory, and we basically interpolate, in this case, what is the entrance time and the exit time of that circle, based on the distance of the radius on the trajectory and the previous location. So we interpolate the entrance and the exit time, and then we calculate how long it takes to go from the entrance to the exit. And this is the first passage time. We do the same calculation for each point of the trajectory, and then also for different radii. So then what is interesting for us are mainly two different metrics that we can calculate. One is the variance of the first passage time along the trajectory and for different radii, because this what tells us what is the scales at which the processes are happening. Basically, I will also show you a bit more later, but when I have a radius of, let's say, 5 meters around the trajectory, this is likely not to change that much. The time that it takes to cross 5 meters, if for this animal 5 meters are very little, is going to be the same, more or less, along the entire trajectory. But when I increase this radius, then I might intersect other movement process. For instance, if I look at 10-kilometer radius, then in some parts of the trajectory, the animal might take very long to live 10 kilometers, but when the animal is migrating, it takes very short to cross 10 kilometers. So if I look at the variance of this 10-kilometer radius across the entire trajectory, I will have a big variance. Sometimes it takes very little, sometimes it takes very long. And so this point of high variance, it's interesting because it means something is happening at 10 kilometers. And then also the slopes of the average time that it takes with the radius also tell us more. But I will show you plots that are a bit more clear in a second. So in this trajectory, for instance, we see for the first part of the trajectory, the animal is mostly, this is just an example that I drew by hand, but there is a first part going straight, then maybe the animal is foraging, meandering around, so maybe around this point and this point, we have some changes in the movement process, and then the animal goes straight again. So again, we can first use a radius that is very small, but then we can fit other radii, and these radii are very big, and they encompass a bigger portion of the trajectory. So when we use these bigger radii, then we are more likely to include different types of movement. Whereas if we use the small one, the blue one, is just taking a very small portion of the trajectory. Finally, if we take a radius that is big enough to encompass the entire trajectory, again, the variance is going to be like null, because there is only one value that takes the entire trajectory. So when we look at the variance across related to the radii, we see that these are all the radii that we can use. Ideally, we use many of them to understand a bit more about the trajectory. And here we have the variance across the entire trajectory for that specific radius. And we see that when we use a very small radius, the variance is very low. And then we cross some points in which the variance becomes very big. And these are this crossing point. If we look at this red circle, a red circle where we intersect, for instance here, a part of straight movement with a part of meandering movement, then the time that it takes to cross these circles that are the same radius, it's very different. It can be very short at the beginning, very high later, and so the variance overall is high. And so we're interested in this point. Finally, we take a radius that is big enough to take the entire trajectory, and then the variance drops again. And then here we have the same information, let's say, but put in a different way. In this case, we look not at the variance, but at the actual value of first passage time. So how long does it take to cross these circles in days? So we have here on the y-axis the first passage time in days, here the radii in meters, and then we take the average across the entire trajectory. And what we're interested in is this relationship. So the relationship between how long it takes and the increase in radius, because ideally you would expect that the bigger the radius, the more it takes to the animal to cross this radius. But this relationship is not always the same. So sometimes this relationship is quite flat, and when it's quite flat, it means that the movement is quite directional. So if it's directional, it means that even if the radius is very large or much larger, since the animal is moving fast, the time that it takes to cross that extra bit of space, it's not much bigger. And so the movement is directional, and this relationship here, it's flat. When the movement is, let's say, you haven't heard the word Brownian yet, maybe you will hear it in a bit. But when the movement is has the same likelihood to move in all directions, this relationship becomes very steep. So if I increase a little bit the radius, the animal keeps moving around in circles, and so it takes much, much longer to cross it. So we can just look at the value of the slope, and when we have a low slope, we know movement is directional. When the slope is high, the movement is tortuous. And this is just an example, again, with Leo, because we know it already. And here we are looking at the plot of the first passage time over time, so across the different years, and using two different radii. So here we have a radius of 3 kilometers, here we have a radius of 80 kilometers, and we know that this animal in his life cycle encounters different or uses different movements. So here, with a radius of 3 kilometers, we see that in all years, it doesn't ever take him very much to leave this 3 kilometers radius. And this is, so maximum 10 days, but more than anything, 5 days. So within 5 days, he always leaves this 3 kilometer circle. So this is probably something that is like his territory, and it just crosses it all the time. But then if we make the radius very large, 80 kilometers, which is maybe something that is more related to the migration, we see that it takes, over the different parts of the life cycle, it can take very different amount of times to cross these 80 kilometers. So here, at the very bottom, this is the migration. So this looks very, it's a similar plot from the net square displacement, but the information is kind of opposite. Because here's migration. During migration, 80 kilometers are crossed very quickly. In 1 day, it crosses 80 kilometers. But for the whole rest of its life cycle, it usually takes very long time to leave the 80 kilometers. And we also see here a difference between breeding and wintering, because in wintering, the animal moves more. In breeding, the animal is very close to the nest, so it always takes very long time to leave these 80 kilometers, so 150 days. But for the wintering, it depends. Some days they move more, some days they move less. And I think, yes, and then there will be an example. So this is about the first passage time. And I think you need to wrap your head around it a little bit of this information, and always think, okay, what does it mean? But it can be very informative. And maybe I would then jump to the code. So if you have any questions about this part, let's talk about it. Where is the mic? Sorry.
Sorry. Where was the question? Yeah.
I'm not sure if this is getting a bit ahead, but I was wondering if there's a good way of choosing these radiuses to check them. If I wanted to identify different scales, that might be different thresholds.
Mm-hmm. No, you will see it later. Ideally, you use a lot of them. And then you fine-tune to what is interesting to you. Then once you find a radii that is very interesting and you think it's capturing the type of movement you're interested in, let's say you want to segment something in your trajectory or classify, then you can use that one to plot this, to have this type of plot and look at the values to classify and say whenever, for this type of radii, whenever it takes longer than this, it's one behavior. Whenever it takes shorter than this, it's another behavior. And this is going a little ahead, because then we'll have a session on classification. But at the beginning, when you know nothing, of course, you can have an idea if you're studying an ant or if you're studying a vulture, of what type of range of radii you're going to choose. But ideally, you take a bunch of them for exploration.
I might just add to that a little bit that in some of the early first passage time, residence time type analysis, people are really interested in like Fauchal and Tvera. They're really interested in like the scales, the most relevant scales. What is that most relevant scale? And obviously different things happen at different scales, so many scales can be relevant depending on your question. But one approach that they kind of liked at the time was to look at the first passage time and see at what radius you have the largest variance in that first passage time. And so that tells you, so if you have a very small radius, then you're pretty much always coming in and out. Not much variability. If your radius is the whole Earth, you're never leaving it. So there's also no variance there. But so like the scale at which the variance of the first passage time is maximized, they'll be like, well, that's the one where the most variation behavior is happening in and out. So if you really want a super automated approach, yeah, there you go. That's the slide.
Yeah. Yeah, no, exactly. That's nicely put. So in the end, when you start with many radii because you just don't know much about the animal, then what you're gonna be interested in is when the movement process is gonna change. Where do I see this jump? At what scale do I see this jump? And then you can focus on those and leave out the other ones. Yes.
This is just a small question. So if your track is leaving the radius multiple times, does that affect how the first passage time is computed, or is it just the...
So if your trajectory crosses the same radius again?
Yeah.
No.
Or like in this example, the second circle on the left,
Yeah.
you have a loop that leaves the radius and then enters and then leaves again.
Yeah.
Does that make a difference?
No, not in this one.
Okay.
We will see how then this is used in the next, in the recurse to look at recursions to a certain radius. But in this case, not, because that same radius is then fit to the next point and to the next point and to the next point. And it's only this amount of time that it's taken. The first time. That's why it's called first passage time, because it's the first time that crosses that specific circle. And then with recurse, like now we look at first passage time in R, and then with recurse we will see how this is calculated again. Yeah.
Yeah. How sensitive is this to fixed rate? Like, is the peak of variance going to change depending on the fixed rate, and then your interpretation of the behaviors would completely change?
Depends how different is your. It's all relative to at which scale your animal is moving and how tortuous is the track and so on. But of course, like this, what is it? Here. I mean, this, if you're fitting this around one point and then you have 3 days before and after the next points, then the point of entrance of that circle, it's gonna be a very rough estimation, because you don't know what happened the previous 3 days. So if you have 1 point every 3 days, this is gonna be pretty much arbitrary. I mean, it's a linear interpolation. Like you take the distance between the previous point and the point around which the circle is fit, and then you look at where ideally it has crossed that radius. So if you have a big gap in between, or the main problem would be then if you have parts of the trajectory in which this gap is very small and part in which is very big. So in that case, you would have to regularize it a bit, or analyze different parts separately.
Yeah, the irregular data always kind of, in many methods, irregular data, are a bit of a nightmare.
Yeah, it's all the choices that we talked about yesterday. Do you subsample everything, making all your estimation equally arbitrary, or do you decide to analyze separately different parts of the trajectory? And you just have to deal with it. Do you have a question?
So following in that sense, when we have this chump data that we have burst of information, it will be better to apply those to the specific burst of information that we have if we are trying to analyze behavioral components, because you will have a lot of information in which you can actually determine first passage time, and then you have a gap of no information. Mm-hmm. It will be better to subsample to those specific areas.
I would say yes,
just,
and then it depends how interested you are in those gaps in between. Maybe you just then keep them separate and fit your different radii to only those chunks in which you have a higher resolution, and then of course you have to be aware that you are ignoring whatever happens in between. So I think all these things are possible, and if you are aware and you declare what you're doing, then it's fine. But yes, I mean, there is no other way around, because then fitting it, I mean, you still have missing information in those gaps. So whatever information you take out of those 3 days of missing data, it's still probably not very informative compared to all the rest. So you just risk to mess up the rest that is more accurate. So I would say you fit it separately. Yeah. Yes. Okay, let's look at the code. So you should have in your folder 4 pieces of code that are relative to the lesson of this morning, 6 A, B, C, D.
D is not yet in there.
A, B, C. D to come. Yes, so we're going to look at the first one that is about net square displacement and first passage time. Loading all the library. Okay, in the scripts you will always find a bit of text, and this is just as a reminder, so you don't have to look back at the presentation, but mainly what is written there is the same that we just talked about. We're going to look at the Fishers again and a Leroy. Drop empty location, all of this we talked about yesterday, and we are jumping into calculating the speed of Leroy. So this is about the autocorrelation we talked earlier, and so we're gonna do a little exercise to see what happens at correlation at different lags. So to calculate the autocorrelation of a variable with itself, if we are interested at the lag 1, so meaning just one difference in time, this is very easy. And so what we are doing there is just calculating the speed, dropping the first location, dropping the last location of the speed, putting them one next to the other. And so we have 2 vectors of speed with a difference of 1, and then we calculate what is the correlation. So very easy exercise if you're interested in a correlation of lag 1, and we can see there that you have a correlation of 0.6, so between one point and the next, the correlation in speed is quite high, which it's expected. Then we could do the same exercise with a lag of 2, and then we see already the autocorrelation is slightly less when we look at the lag of 2. And then this is just an exercise for you to see how you would code it, but thankfully we have a function that does this for us. But just for you to understand what happens, you could do this for looking at the lag between 1 and 100, and then calculating the correlation and the correlation values for the different 100 lags. So correlation coefficient, the lag, and then here we added what is a significant correlation. And then we can see that there is some periodicity, because we see that the correlation drops between 1 and maybe 10, and then every now and then the correlation goes up again, which means there is some periodicity in what the movement process is. And I guess you will see a lot more about this tomorrow, but just so whenever you see this kind of pattern, then something should ring like a bell saying, okay, maybe there is something that happens every day or every this amount of lag for which the animal is going back to a similar speed, because we remember here we are looking at speed. But we don't need to code this, because then there is this function that does this for us, but it's good to know how things are done. And this autocorrelation function does exactly the same. You can specify what is the maximum lag you're interested in. There is an argument in the function that is called lag max, I think. But otherwise, it just takes a default and shows you how the autocorrelation changes at different time lags. These time lags might be irregular. So what R is doing is just taking the value of speed at the observation 1, 2, 3, and so on. If these observations have a different difference in time between them, this is also not shown here. So this is something you should know before applying this function. Yeah, but we would hope that for the first 30 points, they are quite regular, right? the trajectories. And then we can also look at the azimuth and the turning angle, and then plot also using ACF, both of them. And we can see the autocorrelation in azimuth and turning angle is quite low for Leroy. So it means Leroy is going pretty much at a fast pace, but then the autocorrelation in where it goes and how much it turns, it's low. So it's going pretty much in all directions. There is no persistence in direction. And so this is just to show you the same plot that I showed in the presentation. What happens to the overall movement process when we add correlation in azimuth, in direction, so persistence in direction. And here we simulate two trajectories. One trajectory with the autocorrelation of 0.2 in the direction, and one with the correlation of 0.99. And this is just showing you the same that we saw earlier. So when the autocorrelation in the direction of movement is very low, the animal meanders around because the likelihood of moving one direction or the other at every step is higher. When the autocorrelation in direction is very high, if I'm going there, I keep going there, and then I move away from the origin or from the area very fast. And so again, how do we quantify this from the movement process? We have these two methods that I showed earlier, the net square displacement and the first passage time. And so we're going to see how these are calculated in R. Oh, was it par? I just wanted to restore my par defaults. Just to restore my plotting window. So here we import Leo. This is the data set that we used yesterday, to which we added the seasons, the arbitrary chosen seasons of migration and breeding. So this data set, we already cleaned it yesterday. We took the portion of data that were before this time gap that there was there. There was a gap in 2013. So the data is already tidied up, let's say. And then the way to calculate net square displacement is very easy, because we take this function from the SF package. The function is called stdistance and works with spherical coordinates. So we take Leo. So Leo is the move to object with all the locations. And then we calculate the distance between all locations to the very first location. And we squared it. Quite easy to calculate. And then here we can reproduce the graph that I showed you earlier when we look at the net square displacement calculated separately for the breeding and for the wintering. So this is what we showed earlier in the graph, and here there is like a zoom in of the two seasons, the breeding and the wintering. So this is exactly what we saw earlier. We see that there is this periodicity in the life cycle. They usually stay very close to where they were tagged, so our point of origin. Then they move away, they stay away constantly at the same distance, they come back and so on. So this looks, again, very, very ideal. Not all animals will look like this, or actually most will not look like this, because they might be wintering at different years at slightly different distances. They might also not breed at the same time. But let's say for identifying migration, at least, this is very interesting, because you see that suddenly they move very far away from wherever they were before. So looking at this part of the trajectory can be very interesting if you want to classify your track. And here we look at the breeding. So this is in square kilometers. So basically, between June and September, the animal never moves away more than 20 kilometers for the nest. So it's very, very local. It stays around the nest. And then in wintering, we see that there is more jitter. But if you remember yesterday in the turning angle graphs per season, you remember we didn't classify the wintering correctly. So in the wintering, there was a little bit of migration. And here we see it very clearly. So there is this big jump. Ideally, if we classified it correctly, then there would be no no traveling, no high net square displacement in winter. Yes, question?
So I was wondering if we can actually use this to classify the behavior.
Yeah, exactly. That's what I was trying to say. So once you fit this, for some animals looks more ideal than for others, but the value of the net square displacement, it's something you can use for your classification. You could say any time that the net square displacement is higher than this value, the animal is traveling, commuting, migrating. Yeah, for sure. Yeah.
From this graph, we can also say that actually where it's going to winter is a little bit of a limit area, because then it just comes only closer distance. It's never gonna go further away than like it reach almost like a limit. You don't see peaks on top.
Yeah, so this means that the area in which it winters, it's always at the same distance.
Is at the edge.
But it doesn't say in which direction.
Because then it says that it's shorter to the start. It only goes down, no?
Yeah.
In the graph.
So it never goes farther away than this maximum distance from the nest.
So it's like an edge. From this graph, you can already say that it's an edge. It's
the edge.
Yeah, an edge radius, like.
It's like it moves in, I don't know.
So
I'm imagining I have my breeding site, then it migrates, arrives to this new place, but from the new place it mainly gets closer to the breeding site, never further
goes
away.
farther away than that. Yes.
Okay.
Yes, exactly.
So you can read it like that.
The only thing, I mean, most likely animals don't move in circles, right? But let's say this is just a calculation of distance with no context about the direction.
Okay.
Potentially the animal could be, I mean, it's not going to do it, but it could make a circle always at the same distance around your reference point, and it wouldn't show up here. The animal isn't going to do
I understood.
it likely.
I understood it was going to another site at 10,000 kilometers.
Yeah,
was
it
another
goes at
site.
it goes at 8,000
So it's a radius. Okay.
and it never moves farther than that, for sure. In which direction, we don't know. But it never moves farther than 8,000 kilometers from the origin. Yeah. And of course, I mean, especially when we talk about classification, we just have to remember the way we chose the reference point. So it's important to decide that this is a reference point that makes sense for that animal. Yeah. So in this case, it's the first point of the trajectory just because we know the animal was tagged in the nest. But for other animals, you might choose something else. You might choose even the center of the, I don't know, breeding territory if you're just interested in migration and you don't know exactly where is the nest, but you know that they always come back, all the population come always back more or less in the same area. You could say I just take the centroid of the breeding ground and looks how far they go from there.
Yeah, you can also take like the colony location.
Yeah, other locations that make sense for the biology of that animal. Nice. Okay, first passage time. A lot of text here for you so that you always know either the presentation or here, you will have the content. For the calculation of first passage time, we use ADI habitat, LT. And as I was saying earlier, for ADI habitat, we need a metric projection. So we cannot use spherical coordinates because just the package doesn't work with this. And so what we're going to do here, it's a small exercise of reprojection. I'm going to plot Leo again. So this is the trajectory of Leo. And something that I'm sure you will see in the next days, but something we often do to make sure that we have the least distortion in our projection is to center the projection around the trajectory. Right? We take a projection, we say the origin of this projection is in the center of my trajectory, which means the distortion will increase the farther away you go from the center of your trajectory, but if you stay quite close to it, there is no distortion in anything. And here we are interested in an equidistance projection. What happens with Leo is that Leo travels a very long distance, And so what we are doing here is just a small exercise for which we give our projection 2 points. And this is something that Chris told me about last year. And you will see also a smarter way to do this. But basically, we're going to take the minimum, and so the bounding box of this trajectory, and we're going to give 2 points of origin to the trajectory, saying that anything that is calculated between these 2 points has the least distortion. So to do this, we just look at the bounding box. The bounding box is basically making a square around this, and it's taking the minimum and maximum values of the longitude and latitude, and we just take these values and put them here in this projection. So we have 2 points, longitude 1, latitude 1, longitude 2, latitude 2, and we then use ST transform to reproject it to this new projection. To work with ADI habitat, we need a different type of object. So instead of move to, we need the ltraj object. And so we can just use this function as ltraj, and we convert it. It just needs the locations, locations in projected system, the date and the ID. And then calculating the first passage time is very straightforward. We use the function fpt. And then here we are selecting a sequence of radii because we don't know, let's say we don't know the animal. So what we're going to do is taking basically a sequence of radii between 1 kilometer and 1,000 in steps that are exponentially bigger. So at the beginning we sample less, and then, we sample more smaller radius, and then less and less bigger radii. And then here we can also say in which unit you want to calculate the first passage time. Maybe it takes a little bit. Wait. Here we go. So we look at the structure of this object. Big object, because we have a lot of radii. So in this case, the number of, what is it called? The number of rows. No, sorry, what's that? No, this is the list, okay. So here, the R1, R2, R3, and so on, these are all the radii that we chose. And then the length of each of these vectors, it's the length of our observations, because we calculate the first passage time for each location along our trajectory. So now our trajectory is horizontal. Here we have all the locations, and then here we have all the possible radii. So if we only selected one radius, we would have only one element. And you see that at the beginning there are these empty values. And these empty values, the bigger the radii, the bigger the radius, the more empty values we are going to get at the beginning and at the end of the trajectory. And this is because when the radius becomes bigger, the points that are before the point for which we are fitting is not included. And so we have basically no way to estimate the entrance and exit time. So we cannot estimate the entrance time at the beginning of the trajectory, and we cannot estimate the exit time at the end of the trajectory. And so, yeah, the bigger we take this radius, and the more NAs we will have at the beginning and at the end.
So, just to comment.
Yeah.
I have in the past, just because you want sometimes if like an animal is beginning in one area, right? Mm-hmm. And you really want to know when it leaves. And the first passage time will not tell you anything about the first portion of that trajectory. Mm-hmm. Then you can just freely, without any compunction, go ahead and throw in another point halfway across the globe if you want, just to let it come into that area and get that first passage time out. That's totally fair, and it works very well. So that'll eliminate all those NAs in the beginning and the end.
Yeah. Did everyone get that? The suggestion? To deal with these NAs at the beginning? Is saying that basically if you're very interested in the beginning of the trajectory and knowing when this area is left, then you can add arbitrarily another point in the direction from where the animal is coming, just so that we can interpolate this entrance and exit time. And so you don't have all these NAs at the beginning. Yeah, thank you. Never done that, but very fair. This is what I said already. So we have the number of locations here in the dimension, and here the number of radii. Okay, and then we are going to look at what we saw earlier in the presentations. We are going to calculate the mean first passage time, and then extract to which radii the mean first passage time corresponds. And then this is what we saw earlier. So radii against time that it takes to cross these different radii, and then we see that the slope of this relationship changes depending on how fast the animal is moving and how directed the animal is moving. So with increasing radii, on average, it takes the animal longer to leave the circle, but the speed depends. And then this is for calculating the variance. There is another function that is called var log FPT. And if you remember, we said earlier that we are very interested in the points in which the variance is maximum, because that means at the scale at which we are looking, the radius that we are looking at is the scale at which the changes in processes is happening. So first I'm moving very slowly and meandering around, then I'm moving very directly. When the radius crosses these two types of movement, then I have a peak in the variance. And so this is exactly the graph we saw earlier. And so if we're interested in this identifying these peaks, this is how we identify the different slopes. And we have, for slopes we said earlier that is less than 2, we more or less have a directional movement. For steeper slopes, we have a more Brownian movement. And then we are just fitting linear models between the mean first passage time and the radii that we're looking at. Where are the breaks identified? I think it's clearer in the next example. Don't remember how did portion the track here. Yeah, here it's clear what we are doing. So we have the plot of the variance, and here we are identifying breaks. So here we're looking what is the difference in variance. And so here we find these breaking points along the trajectory. And then here we add this, what I said earlier, this is the minimum, and this is the maximum points of variance that we are interested in. So of course there is the biggest hump, that is the biggest maximum. So this is the biggest change in movement process, but then depending on what you're interested in, you can find other points at different radii in which other things are happening. And then we take all of these breaks that we identified earlier. And here, that's why I was saying here it's easier to see. I don't know what was earlier. But here we basically fit a model. And we fit different linear models to different chunks of the trajectory. So we take from the first point to the first identified break, then between all these different breaks. And we fit a linear model to each of these, between the average first passage time and the radii. And so this way we are able to then fit all these different slopes and the points. And so here we can see how the slope changes at the different part. And if we want, we can calculate what is the slope at each of these points, just looking at the breaks in the variance and fitting a linear model separately to each part. But so all of this is quite abstract because we're just looking at the variance. But then if we plot the actual first passage time in days across the per time, so across the trajectory, we can see this better. And this is also what we had earlier in the presentation. So here we're looking at the actual first passage time, so how long it takes to leave a certain radius. and we look at this across time. So how does the movement process changes in time? And here we have, why are these very...
Same plot, no?
Yeah, the same plot repeated. Well, here supposedly you should see something around 3 kilometers, 20 kilometers, and I think 80 kilometers, so the same that we saw in the presentation, and right now I'm not sure why these are the same. Wait. Let's do a little trick. Yeah, here we go. Okay, we'll see why in the script, but we know what are the breaks that are interesting for us. So we just quickly fix it. So we have here a radius of about 3 kilometers, about 20 kilometers, and about 70 kilometers. Can make this a bit bigger. And so here again, we see the different scales at which LEO operates. So 3 kilometers. Here we have the first passage time on the y-axis in days. So we see that a radius of 3 kilometers, it never takes LEO more than 10 days to leave it. But then when we look at 20 kilometers and 70 kilometers, we see that it can take up to 150 days to leave that circle, but sometimes it takes very little time. And so here is where we really see the difference in movement. So just repeating this again, whatever is at the very bottom means that are times of the life cycle in which this large circle, it's left very, very quickly. So in this case, it's migration. So taking about 1 day or 2 days to cross 80 kilometers. But then at different times, it might take much longer time. And so this just it's just a better way to, let's say, interpret what we are seeing, because it can be a bit abstract to just look at the variance. And yes, any question about this before we jump into recurse?
I'm sorry, what did you do to fix the code?
Oh, I will then upload another version. I just knew what were the breaks that were interesting to us, and somehow in the way I ran it earlier, I had a lot more breaks. And here in the code, we just take the breaks 1 to 3. Okay. And in the earlier, when I ran it earlier, I had way more breaks, and the 1 to 3 were very similar. Okay. But we'll fix it, I don't know. It might just be the way I run it.
Yeah, so that's why I also wrote it in the email. Make sure at the end of the 2 weeks to download everything again, because these little fixes, we'll fix it and upload the code. And
I can put this here. I can put this here again, just. These are the breaks we are going to look at. Yeah. Okay, so then I would go back to the presentation to talk a little bit about recurse, which in my opinion is very fun and can be very, very useful. And so the question that came up earlier here, saying what happens, or can we estimate when the animal goes back in the same radius, this is exactly what this package is going to do. So recursions are to just understand where and why animals return, and this can be for many, many reasons. Animals can return to their nest, they can return to their den, they can return to some food patches, or to a water source, for example. And a lot of times we just don't know what's happening, and we are interested in finding these points that are interesting for the animal. And if we don't know where these points are, then we can use the geometry of the trajectory to just find where these points could be. So recursions can be useful then to work on very different questions, to answer very different questions. We can, first of all, identify which are these locations that are most frequently used by one or multiple individuals. We can also then look at when are these locations visited. So which time of the day, which time of the year these locations are visited, how long they stay in a certain area, and also how long it takes them to return to a certain area after they left. And so the intervisit interval is also very interesting, and all of this information can be extracted very easily from this package. Another implementation is when we already have an area we are interested in, for instance, a polygon, it could be a protected area or a colony or whatever, and then we can just give the function a polygon and say, calculate these revisits from that area. So I'm interested to know how often they go to that area, how often do they return, how long they stay inside, and so on. Yeah, and for seasonal, also seasonal patterns and so on. I will show you, I wasn't sure if showing an example of this at the end of the script or not, but of course you can analyze all of this information that we extracted also relative to environmental variables. You will see, because I said this was going to be without context, but I thought this example is nice, so I can just show it, but you will learn way more about the environmental context on Thursday. But yeah, there will be just a small example. And again, the calculation of recurse is very similar to that of first passage time. So we, again, here need regular sampling and also an equidistant projection. This package is created by Chloe Brassis, who also used to teach this class until a few years ago. And now, likely, unfortunately, she doesn't have the time anymore. But she has a very nice vignette with some examples. And also the example code that I will show you later is from her. So how does the recursions package works? It's actually very, very similar to what the first passage time does. We draw, unless we give an area of interest, but if by default what it does is that it draws, again, a radius around each point of the trajectory. So we do this iteratively across the entire trajectory. We have to decide on a radius that we're interested in, and then it will basically cut all the segments that fall inside that radius. So if that radius is visited again, it will show up. But the first time that the radius is crossed, this is the first passage time. So actually, if you fit a recurse, this revisited, I don't remember, the visits function or something like this, the first value that you have is always the first passage time. So you can also use this function to extract it. And then it will interpolate again the entrance and exit time of each of these segments in the radius. So that's why I was saying, again, it's important that there is a regular sampling, and about the projection. All of these things are important to ensure that these enter and exit times are estimated more or less, or as accurately as we can. And so the time between the visit and the time inside each visit is also calculated. So let's look at some examples. Here we are looking at the elephant, and this is the example I will show you also later in the script. That's the name of the function, I forgot. Get recursions, very easy. So here, the way it works is very easy. We have this function, get recursions. We put here this object. It has to be a specific object. Then we decide on a radius in meters. That's why the projection has to be in meters. And then we decide on the time unit we want our revisits to be calculated. And the object that we get out of it is has a lot of elements, so it's quite complex. But this is one part of the object in which you see, this is the ID of the animal, the coordinates, and the coordinate ID gives us basically the point in the trajectory. So all this that have point 1 is basically the first location in our trajectory. And then the visit ID changes, and that means all the times that radius that was fit around the first point was crossed. And if we take the first visit ID of each of the location, this is the first passage time. And we will do this later. And then you have entrance time, exit time, duration of the visit, and a lot of other information. Please, question. Sorry. There. We'll answer all of them, so no problem. We'll try.
Just to understand the visit time, is it it considers the time that it goes out of the radius and come back? Not really.
No, so the visit, so the entrance time that you see here, it's this one. Enter and enter. So after all the segments are identified, all the segments across the circle, then the entrance and exit time is identified or interpolated and then reported there. And then you have two information. One is the time, the duration of the visit, so how long it took between the enter and exit time. And another information is the intervisit or something, so the time that it took to return. So basically the amount of time that they stayed outside before returning.
Okay.
So both of them are in there, the duration of the visit and the time that it takes to return. Was there another question?
So if we want to look at things like site fidelity, we can use a radius of smaller radius circle in order to identify which particular area is revisited time and again.
Yeah, of course. So the idea of this one is a little bit like the first passage time. It's not so easy to use a lot of radius at the same time. So ideally, you will just select some that you think are interesting, but it's a little bit the same. So you can look at different scales, what are areas that are revisited more often than others. And so depending on the radius you choose, you can see something like site fidelity or something like a water source or something else.
Okay.
Yeah.
Thank you.
Oops. Yeah, so here, just an example. In this case, this is Leo. And we, for instance, didn't know where the roosting sites of Leo were. And so the way to find them out was to basically look at first fit the recursion, the get recursions function, and then look at only those points that were revisited more than 75 times. Of course, this depends on how long is your tracking period, but for Leo it made sense, given we had a lot of data. And then putting this filter, we identified just five points that could be potential roosting sites. And another way to also massage this data and interpret them a bit better is that we could look at how the duration of the visit relate with the entrance time. And here we can see very nicely that, for instance, when Leo enters after a certain time in the day, then the duration of the visit is much longer compared to all the visit that Leo does during the day. And if we also add the time of the day, we realize that all these parts, all these late visits are overnight stays, and then these are stays during the day. So it's also a nice way to just separate. And we will see also in the R script later how we can look at visits that last more than a certain amount of time, visits that are shorter, and how we can play around to get different information out of the data. And let's go to the script. Yes, please.
How sensitive are these recursions to irregular sampling? Also
Same answer as the first passage time. Yes.
Okay.
But also the solution would be the same. You could just decide on either a lower sampling frequency for all your data, depending on how big is this difference, or just analyzing them in chunks and just fitting a different recursion function to different parts of your trajectory in which you have more or less regular sampling. Yeah. I also put here the name and the email of Chloe, who made this package, and it's the first package of the list, the package recurse. And then here we load all a bunch of libraries and set the directory on our folder because we need to get the data for these elephants. Here there is a couple of functions for palettes. And here we load the data. We load the data as a move to object, and then we are only interested in one, for example, in one elephant, a biba, and we just plot it just to see how it looks like. This data set was already nicely cut for this example. So we see that the duration of time, here we're looking at the range of the timestamp, and its difference, and it's about 15 days of data. So here we're looking at 2 weeks of tracking of this elephant.
So
the map view? Okay.
maybe the issue is that map view now also uses Stadia and also needs an API that you have to register. So that way it was working yesterday on my computer because I had the API of Stadia registered.
I also have an API of Stadia.
Yeah, and you guys probably don't, and because all this, everybody wants now API key, so that might be the problem. I can't test it. Well, I could, but yeah, maybe you test it. Register in coffee break, whatever, in Stadia, and register the key, and then you should be able to view it. That probably is the issue. You have registered and you don't see it.
We can also see it later and just try to look at
maybe.
it. Like
Maybe in the meantime, I can unregister and see if I have the same problem and then re-register or something like that. I don't
And
know.
you also have the new version of R or the one before that?
I think maybe the one before.
The 440.
Okay.
That's the one.
Yeah. And
We'll figure it out at some point.
But I mean, you can see it. This is what you're gonna see. So, but we'll fix it. Promise. We'll fix it. But anyway, it's just a one line of code because it's the easier way to look at your data if you have it. But if not, you can just plot it. And so so we have these 2 weeks of data, and so you can run probably the next plot. So the next one you should be able to see. It's gonna get the same information. Doesn't matter if you have or not have map view. And here we are basically coloring the trajectory per day. And we already see that there is some sort of periodicity that, I mean, of course we know because we know this data set, that's why we chose it. But it's just a nice way to look at your data if you suspect that something like this might be happening. And so you see that this looping behavior kind of happens every day. So every day the elephant goes out and then back to whatever is it's interesting to him. So here we are using again equidistant projection. This time we don't need these two complicated one with the 2 points because the area is quite small and the animal stays in the center of the area. So we just center the projection, this equidistant projection around kind of the centroid of this location. And this you can do it using the move2 package. The recurse library does not work yet with move2. It works with the normal move object, but there is just this function to convert from a move2 to a move object, and so we just, it's quite easy. We just run that. And we select for a start a radius of 500 meters. And as I showed earlier, it's very easy to get the recursions. You just put the move object in there, a radius of your choice, and the unit, the time unit in which you want your recursions. I mentioned earlier that the object has a lot of information in it, so we just try to look at it. First of all, this object is of class recurse. It has a lot of different informations. So these are the names. There is revisits, residence time, what is the radius that we chose, the distances, time units, and so on. And more interesting than all the rest is, if we look at the structure, is this data frame here that is called revisit stats. So all this it's a list, and so all the elements from 1 to 4, they have just the length of the number of location. So the first one, for instance, revisits, is just an information that tells us how many times each radius along the trajectory is revisited. So the first location is revisited 8 times. The second location is revisited 6 times, and so on. But then in these revisit stats, this is what I showed in the presentation, we have more informations. Because here, the number of observation are the number of location multiplied by how many times each location was visited. And so if we look, sorry, this is what I wanted to show for a better way. First location was visited 8 times. And in fact, if we look at the revisits stats, we see here that this is the ID of the location, 111111, for 8 times. And here we have for the visit ID, the ID of each one of these visits to that same location. And here, she was asking earlier, so we have the entrance time, the exit time. Time inside is the duration of the visit, but then we also have this other information that is time since last visit. So you have the time inside and the time outside. And also the location is comes together with everything. So you see that the first 8 rows have the same X and Y because they refer to the same location. So let's have a look of how revisits are. Oops. And so, yes, we see here that a lot of areas are crossed only once, but then there are some areas that are visited up to 12, 13 times. And if we plot this on the trajectory, we can also see it very nicely that there is some areas in the center that are those that are visited most times. And now I'm curious to know if this will work for you. Let's see, because this relates to also having, I think, a study account. But I will show it anyways. Here I'm just using the function getMap to get a background map for the area of the trajectory, the bounding box of the trajectory. And then I'm using ggmap to plot it on top. So I don't know if this will work for you, but here, I mean, it's the same information. It's just nicely put. You saw a lot of this, I think, with Anne in the visualization class. But here we say the same information. Does that work for you? Yeah. Yeah? Okay. So that's not the problem of map view. You have a question? What is not coming up, sorry?
Yeah, so it is the key, because the
No, but that's the
map-
previous plot. We are to the next plot. The one you should. You run it? The second? Where do you get the error? But where do you get the error?
Yeah, so it's
yeah,
the API
okay.
key that you have to register. So
Yeah,
in
for that you
the
need the API.
in the visualization script, there is at some place commented text, like in the middle of the script, where I put the link how to get a Stadia API key and how to register it in R. So if you follow that, you should be hopefully fine.
Yeah, maybe during the lunch, before or after the lunch break, we can just put up the instructions. So in case you want to do it tonight, you can just follow. So the API key is easy to solve, and then you will get this. The map view thing, maybe it's something different. But the API.
So it's line 83 to 88 in the visualization code. And there you have the instructions of how to do it.
Yeah. Yeah. So that one, I believe you can fix. The map view also, but need more thinking. But anyways, it's just same information put on a background map. And then I mentioned earlier, from the recurse object, we can extract the first passage time. So how do we do it? You can see here all the revisits in the, maybe I go back to the structure. So if we go in this revisit stats and we take just the first visit ID of all the coordinates, only the first one, then this is the first passage time. I mean, the duration of that visit is the first passage time. So here we are taking, extracting the time inside, which is the duration, for only those visits that have ID equal 1. And then if we look at the distribution of the first passage time of a radius of 500 meters, here it's just one radius, we can see that there is kind of a natural break around 6 hours. So it means that there is a lot of locations that are crossed in a shorter time than 6 hours, but then a bunch of locations that takes longer than 6 hours to cross. And so we are interested in looking what is the difference between these groups of locations. So we define here a cutoff of 6, and we plot on the trajectory what are those locations that the animal takes longer than 6 hours to cross. And we see that they are quite grouped in space. So these are all the locations that where the animal spends more time to cross. And then we can also look at, as I also showed in the presentation, we can explore this by looking also at the time of the day at which these visits happen. So here we are doing a box plot of only those locations that, the first passage time at different time of the day. And we see that the locations that takes the longest to cross are those locations that happen at night. So until 3 a.m. and after 15, 16 p.m., then we have these locations that the animal takes longer than 6 hours to cross. And then during the day, the animal takes less than 4 hours to cross all the other locations. So this is a way to look at this, but then other information that we can extract is the overall residence time. So the residence time is the cumulative time that the animal spent in that radius, so at each location, summing up all the times that he went back to it. So this is the residence time in hours. So we see 22 hours at the first location, at the second, and so on. And the length of this vector is the same as the number of locations. So we have just one value per location, and it tells us all the times over the tracking period that the animal has spent there. And this looks quite different. So we also have some sort of break here around 20, and we have definitely some locations where the animal has returned a lot, or I spend a lot of time overall, like more than 20 hours time per location. And we can also map this. And we can see that this looks quite different than the other one. So the first passage time had these blue areas at the margin of these loops, whereas the overall residence time is here in the middle. What does this mean? That there are areas that once the animal enters, takes very long to leave, and this is because they were overnight stays. But then there are areas that maybe the animal spends every time very short time, but overall during the entire tracking period, the animal has visited many, many times. So the overall time that it's spent here is very long, even though each single visit is short. So it's just a different type of information. And in fact, if we look this time, not at the first passage time, but at the overall resident time, we see that the pattern is opposite. We look at the resident time per hours of the day. And if you remember in the previous plot, the areas where it spent longer, they were at night, whereas these areas where the residence time overall is long are during the day. So you can imagine it's whatever resource the animal needs. So it returns to it every day. Every time it stays short time, but it visits it every day. And overall, it sums up to many hours. And so we looked at first passage time. We looked at overall time per location. and then we look at the number of revisitations that we haven't looked at yet. So the time since the last visit, it's another information that is in the this time outside, basically, how long it has passed between consecutive visits. And we also here see that there is most locations are revisited almost on a daily basis. And then there are some locations where the animal returns basically after a week. So it takes the animal 8 days before returning to that same radius. So this we can also just filter them out. So returns after 1 week are all those revisit that happens over 7 days, and then we can also map those. And this is only one portion of the study area, basically, where the animal returns every week. So I think all these maps that we showed highlighted different locations on the map that might be interesting to the animal, but for different reasons. Some can be overnight stays, some are resources that they need during the day, and this is an area where they return every week. No questions? And then this is for shorter revisitation time. Yeah, and this was just, so here I'm just zooming in all those revisits that are shorter than 1 week. And we see that also there is some sort of periodicity. So there are places that are visited like over time. And so here we're not doing any periodicity analysis, but this is again, like we saw earlier this morning, something that could ring a bell and say, okay, maybe it's interesting to look why the animal is going back over and over again. But then you would do other type of analysis. And maybe, how are you? Should we do?
We have tons of time still. Maybe finish recursion?
Yeah, and then we take a break. How do you feel? We can finish this section on recursion, then we take a coffee break. This is just an example to pick a smaller radius. Just same function again, and then we see how the revisits change. And then if we pick a smaller radius, we can see that the locations now that are interesting become a more point in time, so we can maybe identify 4 areas that are now of interest, whereas when we picked, of course, a larger radius, we had a bigger area. So then again, to answer one of the questions that came up earlier, of course, you can pick a different radius and decide on what you're interested in. Like, is it a very specific area? Is it the roosts, or is it the foraging patch? And based on that, you will decide on your radius. And I don't have an example for the polygon, but this was just for you to show that you can include a polygon. I think it's still a SP object, so it doesn't work with SF yet. But with the get regressions in polygon, you also can get all the same amount of information, but for a specific area that you define, rather than all the points in the trajectory. And now this part is going to be a little bit of a spoiler for Thursday, just a short one, because of course we mapped all of these locations in the area, so it comes natural, like what's happening in that area. We're curious to know why the animal is returning there, and most of the times you're interested in what's happening in the environment. So we have this idea that, of course, if the animal is returning there every day, there must be something in the environment, a foraging patch or something that brings him to go back. And the environmental layer that I have here doesn't give us a definite answer, but it's just an example of how you could approach this. It is very easy to extract, to match all this revisit information to the environmental layers, because of course they are in the same order of your location. So you can just use your location of your move to object to extract the environmental information of those locations and then match them back with the revisit. They are in exactly same order, same length. So here, Here we see that the number of rows in the Habiba move to object is the same as the length of the revisits. And also, if we look at the revisit stats again, we see that here the X and Y columns are also retained. So you have all the options to match environmental information. And in the data folder, we uploaded a raster layer that is about NDVI. You will learn all about NDVI on Thursday, but it's basically an index that tells us about the health of the vegetation. In this case, I mentioned this already a lot about the reprojection. We have to make sure that the projection of the environmental layers and the projection of the locations match, and this is not the case because now NDVI here has a UTM projection, so we need to make sure that the points match the projection of the layer. And again, this you will hear it a lot, but as a rule of thumb, you always reproject the points, not the grid. And the explanation for this will come. But so what we're going to do here is that we reproject the Habiba to the projection of the raster layer. So as a sanity check, do they overlap? Yes, we can plot them one on top of the other. So most likely they're in the same projection now. And we can also now plot the revisits. Where are they? Yeah, we're coloring here by residence time. And we see that there are these areas that are the most visited one in the center. And of course, then what you might wonder is that we have the layer at the bottom, we have the revisits on the top, and we are just going to pinpoint what's happening on the layer underneath, and we are going to just extract what are the environmental information at the point in which the revisits are happening. And this is very easy. There is a function from the Terra package that is called extract. So we're just going to extract the NDVI information at the location of the Habiba, and we're using also an interpolation method. And as I said again, unfortunately, this doesn't give a definite answer to our question to why they're coming back, but we have a little bit of a hint. And here we are looking at the, if you remember, there was this cutoff, and there were areas that they were revisited many times, so more than 20 hours of time spent at the location. And now we can plot the NDVI why it doesn't show the false pose? Here you should have two plot. It's always something unexpected. Because then I rerun the recursion again. Wait. Moment. Moment. Here you go. Sorry, because I ran the recurse at 100 meters, and so all those locations were not existent anymore. So here you have the utilization when it's higher than 20 hours, then the NDVI is higher. And the higher the NDVI, the healthier the vegetation or the more abundant the vegetation. So basically areas where they spend overall across the entire tracking time more time are areas where the NDVI is also higher. And we can do also the same per time of the day. And so we see that the locations during the day are locations where on average there is more vegetation or more abundant or healthier vegetation. So they probably go there to forage. And the mistake happened, if you want to correct your script, because here I ran the recursions at 100 meters, giving it the same name. So maybe we can just change this name here. And now it should be fine. Any question before we have coffee? Please.
You said that for the Terra extract NDVI function that it's also interpolating. What did you mean by that?
It's interpolating because basically, I mean, I don't want to spoil too much of what's going to be covered later, but basically you have all your pixels of your raster, and depending on where the point is in that pixel, if the pixel is very big, you don't want to assume, you could just say take whatever value is underneath, but since the pixel might be very big, you might want to say, okay, if the point is closer to the margin with another cell, I'm gonna interpolate between the cell underneath and the next cell, so that you have something that is a bit more realistic because the environment is not pixeled. So it's just a way to have a more realistic overview. But depending how fine is your resolution, if the resolution of your raster is very fine, then you can just take the point underneath. But bilinear interpolation is often used for this. This is going to answer some of the questions that came up yesterday when we were talking about what happens with GAPI data. We have irregular sampling. What can we do? Should we always subsample? Should we interpolate? Hopefully this is going to answer some of the questions that came up yesterday when we were talking about what happens with GAPI data. We have irregular sampling. What can we do? Should we always subsample? Should we interpolate? Hopefully this will be a tool that you will be able to use to kind of deal with GAPI data, because this method is supposed to work and help us to estimate what is the probability to encounter the animal in between locations. So in the time gaps in which we did not observe the animals, based on the type of movement that it's doing, because we mentioned yesterday, if you remember, that if the movement the animal is very directed, not so tortuous, it's quite easy to predict where it's going to go next, because it will maintain this direction. Of course, if the movement of the animal is more tortuous, then it's a bit harder to predict where it could be. And so taking into account all this information, we can then estimate the probability of where the animal could have been, when we did not observe the animal. So for this, we're going to look at the variance components of movement, and I will show two methods, let's say. They are implemented in the package move, not yet in the move tool. So we'll look at the old package move, and as I was saying, it can deal with irregular GAPI data. And the way overall both of these methods work is that we have our trajectory. We're going to omit one location of our trajectory. We're going to look at the straight line, connected the previous and subsequent location. And this distance between the estimated and the observed point is going to give us a measure of how hard it is to predict where the animal was. Yes, so let's look at two examples. Here we have on the left a more tortuous trajectory, so with steeper turning angles, and then a more directed trajectory to the right. And so we can, again, as I was saying, we can just omit one location at a time, draw a line connecting the previous and the following location, and look at this distance. And we see that in the case to the right, the trajectory is more directed, so the distance between the estimated and the observed location is not so big. And in the other case, it's bigger. There is a larger distance. So based on the variance that we calculate, this variance is this distance that we have between the predicted and observed location. Based on this variance, we are able to calculate the probability of where the animal could have been in between these gaps. And the good thing, this is why this is called the dynamic Brownian bridge movement, is that this variance is dynamic. So we are not assuming that the animal is going to move the same way along the entire trajectory, but actually we allow this variance to change in different parts of the trajectory in a certain time window. And then we will have to decide on the time window, but we will see that this does not really affect so much the result. So, of course, this will also take into account how big is the gap between our location. So the smaller is the gap, the more consistent or directed is the movement, the smaller will be the probability area around the trajectory. The bigger the gap, the more tortuous the movement, the bigger would be the probable area in which we could find the animal. So this is often the case when we have a low sampling frequency, low correlation in the azimuth, so tortuous movement. This is a more directed movement. And this is the case in which we have very high sampling frequency. And I think this is very interesting because we will see later, this afternoon with Anne, how the dynamic Brownian bridge is used also to calculate a measure of space use. And oftentimes, you will hear a lot about this, but oftentimes this is considered like being a home range. And this has nothing to do with being a home range, because what happens here, for instance, is that if we have very, very high resolution, we know where the animal is basically at all times. And the probability around this is very, very small, which means virtually we have no area around this trajectory, which doesn't mean, obviously, that the animal didn't use any area, but it just means that we are so certain about where the animal is that there is no probability area to calculate. So this really has to be treated like an estimation of the area used, or where the animal really occurred during the time. And the higher, the more certainty we have, the smaller this area will be, because the more certain we are about where the animal is. So I think it's just very important to keep this difference, but you will hear more examples about this. Yes, so let's look at these variants of DBB in Leroy again. We know that Leroy is a nocturnal animal, so Leroy moves during the night and most likely sleeps during the day and is in his den. And so we can see here that the variance is much higher during the day, during the night, because it's moving around, whereas it's very low during the day. And it's because during the day it's in the den, and so we are quite certain about where the animal is, because it's quite stationary. The only, wouldn't say a problem, but the only caveat with this type of variance is that when it's easy to predict, the variance is always low. But it could be easy to predict because it's stationary, or it could be easy to predict because the movement is consistent. And this would give us, in both cases, a small variance. So it's just a measure of predictability of the position, basically. So a little bit, something that could be a way to kind of differentiate this is this dynamic bi-Gaussian bridge movement model, because here we will not look at just the distance, the straight distance between the observed and estimated point, but actually at two components of this distance. So we will have an horizontal distance, that is the sideways component, and then a forward-backward distance. So the first one, the sideways, it forms us more about the variance in the direction of movement, whereas the forward-backward about variance in speed. So how is it like more advanced or backwards compared to where we expect it to be at a certain speed? And so if we look at kind of a similar plot, again per hour of the day for the fisher, we see that there are kind of three options. Values could be around 0, and around 0 would be that the animal has the same likelihood to move in all directions, and the same likelihood to go sideways than to go forward and backward. And usually this is, the case in which this is most common is when you have a lot of GPS error, because otherwise usually you tend to have values that are either positive or negative. When you have positive values, it means that the movement is more directional, so you have more forward-backward component. And when the values are negative, it means that it's more side-to-side. And so here we can have kind of a more, another level of differentiation of the behavior, for which you could have more directed movement at some time of the night, and then a more maybe search behavior or sideway component at other times. So it just gives us an extra layer to differentiate these types of movement. And then we would look at this in R. Do you have any questions? No, crystal clear. Crystal clear, I said. So we can look at script 6C. Load all the libraries. I was saying that to calculate dynamic Brownian bridge, we have to use move package, not yet the move to. So here we load the file using the function move. And as in the methods I showed earlier, we need an equidistant projection and a projection that is centered to the center of the trajectory. So we use this SP transform to center it to the centroid of the track. And then applying to calculate the Brownian motion variance, it's quite easy. We have here this function. This will take a little bit to run, so meanwhile I run it. And then here we can include what is the error, the amount of error we kind of estimate for our GPS. This will depend on your devices. And here we have to specify a window size and the margin, and you will also hear more about this afternoon, when with Anne you will use dynamic Brownian bridge to measure space use, utilization distribution. But for now, for you to know, the result is not really affected so much by this. The margin is something like the NA we saw this morning in the first passage time. So the margin are just some values at the end and at the beginning of the trajectories, in which we just omit somehow the calculation of the variance. And then the window size is because we have this leave one out location. We do it in a certain window, and then we move this window. And the dimension of this window is kind of what gives us the possibility to have this variance being dynamic. So in some part of the trajectory, we expect this to be more directed, in some other part to be more tortuous. And depending on the sampling frequency of our animal, we could decide on a certain amount of locations that we want to use to do this variance estimation. But you will see it's not very affected, and in many cases you can use the default values. And it finished calculated, fill the time. So we extract the variance from the Brownian motion that we calculated, and then we also create a data frame with the hour of the day, and here we recreate basically the plot that we saw earlier. So it's quite easy. Again, you see low variance during the day when the animal is in the den, most likely, and then higher variance at night. Yes, please.
Sorry, you mentioned the location error is dependent on the device. Is that,
Yes.
what are the units on that? What are the units on
in
location?
meters.
In meters.
Yeah.
And how do you sorry? Okay,
Yeah.
for sure. And how do you estimate that?
Oh, usually this is something you should know from the device.
Okay.
There are ways to, Chris, I don't know,
So
maybe Chris will
CTMM
talk about
style,
this.
like
Yeah, exactly.
stationary,
so this is kind
stationary
of a,
data?
Yeah, this is kind of a rough way to do this. There are better ways, and if you also collect some stationary measurements, then
Yeah.
you can empirically measure what's the actual error of your device. So not
Sure.
what the company tells you, but what you actually see. This is just a way to add this information, but we are assuming that this information is constant along the trajectory, and we are basing it on.
So actually, we don't do it because it's like easier to just like put one value, because we often don't have it. But if you have, so with CTMM, you can actually calculate the error for each location.
Yeah.
If your tag also has the needed attributes to do that. So you can calculate the error for each location, and this vector of errors you can enter in the argument. You can do that. No, it's not what normally anybody does, but you can actually do that. So, and that is really cool because there will be errors with really large. There will be locations with really large errors and other locations with really small errors, and all of that can be taken into account. So just.
Okay. That's nice. Yeah, because then your animal will cross area where satellite reception would be lower, and other place in which it would be higher. I didn't know you could include the whole vector. So this is the rough way. Yes, and just another short example to reproduce what we saw earlier for the dynamic bi-Gaussian bridge movement model. It's a slightly different function, but it works the same ways. Again, we have to define window size, margin, location error. This will take maybe a little longer.
And the window size is smaller here?
Yeah, 31. I don't think it's necessarily has to be smaller. Do you know why this case?
It's different functions, so it has different defaults. I think it's basically because it's the bi-Gaussian, isn't it, what you're doing?
Yeah.
So it's the bi-Gaussian variance and the Brownian bridge variance. It's two different functions. So they have different defaults. I'll talk about the defaults this afternoon a bit, but yeah, don't worry about those numbers. That's the bottom line.
Yeah, I don't know exactly why this is lower than the other one. But the second one I never used myself, so I don't know. Know the theory, more or less.
What does these stars mean actually on this plot?
Maybe the outliers?
Oh.
I would say, those that are just very far away from the quartiles. I think so, yes. Nothing to do with significance or any of this, no. Here, look. Here we can decide in the box plot, you can decide how you want the points to be shown, and we put a star. You could put anything else.
Are we all meant to get the same plot?
Yes.
Interesting.
Okay. When did you load the script? But I mean, I didn't change the source data.
Well, when we started this.
No, but you have the same plot.
Yeah, the same. But why I don't have this horizontal line and he has this horizontal?
I think it's a GG RStudio
Yeah,
thing.
13 is clearly different. Like 13 here and 13 there. It's not the same thing.
No, not the same.
Randomness doesn't stop.
The line is here. I don't know. And you all run this BGB variants? I mean, the second plot? We're all looking at the same line 64? Okay. I do not know. I mean, we're all looking at the same file, I believe. And I don't think there is any sampling going on in here, no? Any randomness in this? No.
No?
The three of us have the same.
Did you change
I uploaded it this morning, but they said they loaded it this morning, so the script.
But the data, you didn't change data from yesterday.
No,
from
so the data,
yes.
so actually the data that is loaded is an example data set within the move package. So
So the data, so if you have a Leroy saved from yesterday in your environment or from this morning, this is different.
yeah, so if you haven't loaded, so because we do stuff to the objects, you might have like just 1 day of data or just 2
Yeah.
months or whatever. So overwrite it. So load again the date. That might be the problem, that you use an old Leroy and not the
Yeah.
full data.
The folder data,
Yeah, so, yeah.
No, so
it's just in the one, the objects.
No, what we are trying to say is that please run this line, all of you, even if you have a Leroy object already in your environment, reload that, because we used this Leroy several times and then we did things to it. So just make sure.
things
I
are
don't know.
happening that are not explainable.
I do not know. But we can check for that.
For
But
years it has been this plot.
yes, it never changed. Leroy has been doing the same all these years of AniMove teaching, I think.
Leroy is sick of doing the same thing every year.
Yeah, so one solution, because if you're running like one lesson after the other, and you're maybe overwriting stuff or not, and objects might be called the same, and then you do miss a line, and it doesn't give you an error. The way to solve this is to if you go to session, you can restart R, and then start the script from the
Fresh,
beginning,
yeah.
and then hopefully
it will
look the same.
But yeah, in any case, I mean, the interpretation is the same we talked about earlier. And here is just for you to have this piece of code to do it in R, but it's not, yeah, it's just a few lines of code. It's more about the interpretation, to be honest. And I would be done with this part, and then I would pass it on Anne for a case study on learning from sampling frequency and how this affects your variables. So we heard a lot about this already, so she has a brief. Is it okay with mine?
Yeah, please. Anyway, yes, we got our data. So the reason why I'm downloading it directly from MoveBank and not using the file that we were using yesterday of the bats is because yesterday we were using a file with less attributes, and we need, for this example, more attributes that are actually on MoveBank, so that's why I was downloading it again. So as we saw yesterday, from the trajectory, we can calculate different things. So here, we're calculating, oh, I see, this is the old script. It's okay. Now you can actually set the units in the speed and time lags in the distance function. You can actually set which units you want. This was not possible in last AniMove, so I oversaw the script, so we did it like this. But both option works. The other one is just easier because it's within the function already. Then we can do a summary table. So the first thing, I want to group my data. Let's say, for example, per individual independently. If I have my tracks per maybe deployment, but I want like this summary table, I actually want per individual. So I look, how is my column call that is defining the individuals. And then I just group by the individuals and also by the sex. So we get a nice summary that shows the mean speed, the mean time tracked, and the sum of the total distance traveled by each of the individuals throughout the entire tracking time. With these few things that we calculated, we already can use them to maybe find differences between individuals, differences between groups of individuals, and just doing simple comparisons between the groups. So as the next methods don't like factors, we just, no, I know what the drop levels is. So MoveBank has female, male, unknown, and there's this extra level, and methods that we'll use in a second don't like if there is a level that is unused, so we drop the levels that are not used for all factors included in the table. And let's just have a look at the cumulative distance that each individual has traveled divided by the number of days that the individual was tracked. And we can see that there seems to be a difference. Well, there could be a difference between males and females. So what do? We do a t-test. So the t-test doesn't like units, so we drop the units. And what do we have? Not significant, so there doesn't seem to actually be a difference. I mean, we can also see this from the box plot that there might not exactly be a big difference, but we test it. We can also test it by just doing a GLM with the advantage that in this case we can add more covariates to account for different things. So one thing that we always have to do if we do a model, we have to check if the assumptions are kind of met. And we can see the QQ plot is kind of okay, the residuals, but it's telemetry data, so we can't expect like a perfect fit of it because they're very irregular. If we look at the summary of the model, actually what we see is yes, sex is not different, but what is actually significant is the time tracked. And of course, if you have one individual that is tracked like over 1 day and another individual tracked over 50 days, it does make a difference. So the amount of time that an individual is tracked is always, for whatever you're doing, it's really, really important that you know how long an individual has been tracked and the differences between your individuals that you're using, because it influences everything. The duration of the track is something that you cannot ignore. And now let's have a look if there is, for example, a difference between speed in males and females. And here the difference seems to actually be larger. So we can also test if there are actual differences between the means. And yes, it's the difference between the median speeds between females and males seems to be different. When we apply, we can also do a GLM. And if you remember from yesterday, speed and distance always has this long tail. And so these kind of data need to be transformed. Mostly log transformation or square root works. But if you don't know, like, I mean, you can start testing different transformations, but there's a really nice function, the boxcox, which actually gives you the optimal transformation for your data. So when the likelihood is the maximum, this is the exponent to which you can transform your data to normalize it. And so we can see here that in this case, it's 0.2. If you would have something around 0.59 or 0.51, just use a square root. It's those little decimals don't make a difference. It's easier if you just do a square root. It's also easier it's more straightforward also to explain. But if that is not the case, you can just use this as a transformation. And here you can see time tracked is not any more significant. And what puzzles me, because yesterday evening, sex was significant, and now it's not. It's these random things that are happening this week, which I can't explain, but it used to be significant. And I've run this now. I don't know what happened. Anyway, so the sex was significant.
But maybe just to add a little thing, we are just showing this. I mean, you wouldn't apply a GLM normally in this case. This is just to show you like some pieces of code of, in case you need a different family, you can use it. I mean, GLM is generalized linear model, and then later you will see an example with GAM, generalized additive models, and it's just so you have these pieces of code in case you want to use it for something. If you're just gonna analyze speed and time tracked, and you are normalizing your data to fit the Gaussian distribution, then you don't need any other family. You can apply a normal linear model, but just to show that it would be the same, just changing the name of the function, basically.
And now, do you remember yesterday what we said about speed? That speed is highly affected by the fixed frequency. So different fixed frequencies have different speeds. So now, if we actually look at the mean of the median, just to get it over, yes?
Speed estimates.
Speed estimates, sorry. Yeah, the wording is often really important. So here the difference is not really big. It's the average fixed time for males is 8, and the average for females is 10 minutes. But it might be large enough that you actually find the difference in speed between sexes that is an artifact of the data and not a biological feature. So always when you do analysis, think about what is actually biologically relevant. If the difference that you find, even if you have a significant p-value, think about it. Is it actually a difference that is biologically explainable and relevant? So I have several examples of that has happened in the past. A colleague of us here, they were measuring tarsus length of bats, and they found a significant difference between males and females in the species that don't have morphological differences. They were like, is this like, so we went on discussing is this like biological relevant or not? Because the difference was significant, but it was a fraction of a millimeter. So is this biological? So you really have to think about it. If statistic tells you something, but then you have to always think about the biology of the animal. Another example that in a project that I was working on, we were working on calculating ODBA values of bats. You will learn more about this maybe on Thursday. And then we found differences between years. And it's like, are animals moving more in different years? It's just this is like really weird. So we were lucky that the animals were tagged with different tag generations of the same manufacturer in different years. And actually there are differences in the sensors of the ACC built in. So we found, so if we wouldn't have thought about it, we found different energetic expenditure throughout the years, which was not true. It was just an artifact of the device that was measuring the ACC. So just always think about what you're finding in your statistical results.
Yeah, and maybe just to add on that, I mean, saying the same things in other words. Like, of course, we want to be surprised, and it's also nice to see that things don't meet our expectations. But if they don't meet our expectations, then maybe let's make sure that let's try to take into account all possible other factors in the way we are measuring things that might be the cause of that. And then if this still holds true, then, we are happy to be surprised and to have things that don't meet expectations. But let's first be sure, like in this case, why are they spending more energies during different years? And of course, you could go on thinking, okay, maybe the wind was different. Maybe the next year the wind was on average weaker and they had to flap more, so they're spending more energy. But before doing all this, thinking of what could have been, look at your data and look at all these other things like tag generation, for instance. So it's just a reminder to use these anecdotes to remind ourselves to check our data.
And also, just because the p-value gives the significance, always use the, I forgot again the word.
Effect size?
Effect size, thank you. It just doesn't stick with me today. So look at the effect size, because if the effect is really, really, really small, it might be significant, but is it relevant? So it's just something that you always have to think about, because it's really easy to do a model and to say, oh, look, this is significant, this, but actually, we always have to always, always, always have to keep the biology of the animals in mind and think about if it makes sense or not. And here, just for you to have the code with which you can then expand on for your needs, we included different examples, how to include complex correlation structures, how to include random factors, how to choose or how to be able to state different families, and how to do a model that includes smooth terms. So now we create another table that includes also time. And if we look at the autocorrelation structure, we can add it so we can decore this argument here. So this is what you have to do to then afterwards add it to the core. AR1 is one way to add the correlation structure to the model. So which value do you choose? So the way this works is you have to look at the autocorrelation of the data, and you take always the value of the second lag, well, of the first lag. So the value of the first lag, so here's about 0.2. So we say 0.2 here. And this will give, the model will account for the autocorrelation within the data. And now if we run the model, and here we have stated the autocorrelation, the correlation structure of the data, we can see that the residuals have hardly any autocorrelation. So we've accounted for it within the model. This is just using an LME. If our data are not normally distributed, or we don't transform, or we can't, or well, they have another structure, and there are families that fit for that structure. We can also use, for example, the function DLM PQL. There are many different packages that have different functions, and this one works nicely. Maybe you can work better with other ones, but we choose this one as an example for a generalized linear model. And again, we added the correlation structure in here, and also the random factor, so the individuals. The differences between individuals are counted here as a random factor. And you can see again, by adding the correlation structure in this model, again, the autocorrelation in the residuals is also not there. And then the GAMs are really useful when you have covariates that don't have a linear relationship with your data. So for example, you can account for time of day, which is not linear, it's circular. Or day of the year, which is also circular. Or space. You can include latitude and longitude to account for things that they encounter in space that are not structures or features that are, and the relationship is not linear. You can include temperature. You can include many different things where maybe there is not this linear relationship. In this case, because we're only looking at sex, that doesn't make sense because I mean it's a linear relationship, and this model always has two parts. So it has the GAM, which is the smoothing, the model with the smoothing term, and then because we have then a mixed GAM, we have added the individual as a random factor. You also get the linear model, which is capturing the. Now, how was this, Martina? The LME and the GAM.
There is one part that explains the random effect, and then the one part that it just looks at the smooth effects. And in this case, we don't have any smooth factor because we did not include it. This was just for you to have a little piece of code of how you would apply a GAM, but this is exactly not the example in which you would apply a GAM.
And one thing that, because we always have the tendency to look at the R square, in GAMs, it's normally really low. So even if the model is fitting well, the R squares in the GAM models are usually pretty low. It's not the best measure to see the fit of your model. Let's put it that way. And then it's basically the same. And then we can also, yeah, Martina?
No, just because this is the end, no?
Yeah, this is the end. So just here again, you have the checking the assumptions of the model. So here we can also see that even though the R square was really, really low, the model actually fits pretty nice. You have a pretty nice QQ plot, and the residuals are kind of normal distributed. So, yeah.
No, I just wanted to add that we did this exercise of including the autocorrelation structure. And we said already the autocorrelation is just intrinsic in movement data, and the more finely we sample data, so the highest the resolution at which we sample, then the highest the correlation, and you will learn a lot about this tomorrow. And autocorrelation isn't bad, it's just natural for this type of data, but this type of statistics is not made for working with autocorrelated data. So that's why we need to account for it if we want to apply these methods. And so data are assumed, samples or observations are assumed to be independent in time and in space and between individuals. And so we need to account for pseudo-replication between individuals and autocorrelation structure in time and sometimes in space in these types of methods. Then there are other methods for which this isn't a problem. But in this case, so just for you not to look that, oh, autocorrelation is bad, we need to remove it. But for these types of methods, you have to, otherwise you're just replicated information. So the information is assumed to be independent. If you have the same information repeated multiple times just because it's one step after the other, that this isn't adding any new information to your model. So you need to account for this.
Yeah, so without autocorrelation, you don't have movement. Yeah. So it's the problem is that these methods have been developed before with other kind of data. So we're trying to squeeze in our movement data into methods that are not thought to be dealing with these methods. So we have to account for it, and probably even though we do everything, we are violating assumptions of these models. There is hardly a way around of not doing that. So yeah. But Chris will present how to embrace autocorrelation of movement data, like how to actually not to drop like 90% of your data to apply a model, but actually to embrace the autocorrelation and to do some, and to use all that information that is in there. Questions? About other parts that yesterday, I don't know, whatever doesn't have to be related to this little example, but to other things that are going around in your head.
Oh, okay. Sorry, just an ignorant question about the correlation structure that you're adding here. Is this accounting for space or time or both?
Time. This is just accounting for time.
Just for time.
You can also, so one way, for example, is to account for there are different ways to account for autocorrelation. One way, for example, to account for autocorrelation in space is doing a GAM and including latitude and longitude, the coordinates, as a smoothing factor. And that will account for it as well. So you don't necessarily obligatorily have to add the correlation structure. You can also add the variables to the model to account for the autocorrelation.
Okay, thank you.
Yeah, Chris has a way better answer probably.
So when you have tracking data, even though they're locations, for the tracking data, you don't have to correct for spatial autocorrelation because it's not a spatial field. The tracking data is a time series. The argument is time. The output at a given time is location. So when you talk about correlation, you're talking about what's the argument. So you're only concerned about temporal autocorrelation. If you have spatial data like a NDVI, then the argument is location and the output is the vegetation. So in that case, you have to worry about spatial autocorrelation. And the only time you have to worry about both is if you have a dynamic spatial field, where you talk about vegetation at a certain time and a certain location, and you can have correlation in both time and space.
Thank you, Chris. That was better than my answer, definitely.
I'm good. It was related to that.
Mine is not related to that. Okay.
No.
Okay.
Yeah.
I just wanted to ask, you talked about the track duration and how it's really important to account for it, but is that in sort of every context, or is that very question dependent?
It will be question dependent, but always think about it.
Okay.
Don't like, because if you're mixing tracks that of 1 day and of 3 years, just, think about what you're doing, because yeah.
There will be anyway differences related to other factors. In that specific case was cumulative distance. I think it was cumulative distance. And so that's why it was extremely important. But in general, still you might, depending on how big is this difference of, as like this extreme example of tracking for 1 day or tracking for 3 years, then you still might see a lot of differences in other values. I mean, if you're averaging through a day or through 3 years, there will be.
Yeah, because the longer you track, the more rare events you capture. And then you can actually, so if you have a long tracking period, you can have like depending on
or
the
difference
species,
in the life cycle. I mean, migration, local movements, and all of this also.
But you kind of can have an average that is kind of stable, maybe. But if you only have a really short period of time, you don't know if is this what the animal normally does, or is this just in these 3 days it was doing something very specific, or it was raining all day and that's why it didn't go out, or I don't know. So you always have to take into account when your tracking is happening and if it captures a long enough time period to actually be compared maybe between individuals. Because the danger is if you compare between individuals and the individuals have different tracking durations, they might capture different times of the life cycle, different behaviors, and then you're not comparing actually the total of individuals, just like sections that are not comparable. So you just have to think. So it's not that you can't do it, it just depends on your question, but always think about it. If you maybe want to say like, okay, if you're using a bunch of individuals, it's like, okay, I want to use individuals that have a minimum tracking time of 1 year. Everything that is below, I just ignore, because you have enough data. Of course, if you have like, your 10 individuals and they're really different, you try to do the best out of what you have, but always take it into account that this will affect, it will be like, we will just get results that are not representing anything, just the differences in data, not differences in behavior, differences in the way that the animal uses the environment or space or what,
And I mean, there will be questions that you can't answer with your data set. That's also often the case. I mean, that's why we need to think of all these different effects, and then ideally you would try to, plan and schedule your devices according to the questions you're going to ask or you're interested in. Sometimes we don't have this chance. Sometimes we just get data collected from other people for other questions, and then we need to do the best out of it. But still, at some point, we need to accept that certain methods we might not be able to apply to all types of data. So some data or some parts of the data are good for some questions, and some other parts for some other questions. And this is just how it is.
Yeah, and there is no magic happening. Even if methods say like, with this method you can like, I don't know, do like you will get a really nice answer. As if your data doesn't fit, it doesn't fit. You can't like with the interpolating. If you don't know where the animal was, whatever you do, it can be really brilliant methods that kind of estimate where the animal could have been. You might be less wrong, but you're still making it up. So it's like, it's just things that you have to think about, especially to be able to interpret the results and to make the correct comparisons and to draw the right conclusions. You have to take into account all these things, because each data set is different. It's so heterogeneous that you always have to keep that in mind to draw the right conclusions. Because if not, you might be just the noise. You might be just drawing conclusion of noise, and that is not. Good.
Ready for lunch?
Lunch? Okay.
Then we start here at 1:30 again.